It is my pleasure to talk to you today about vaccines and monoclonal antibodies for HIV prevention. I think we are all well aware of the scope of the ongoing pandemic, and I simply want to call your attention here to the last couple of points, reflecting that nearly twice as many individuals acquire HIV each year as those who die of AIDS-related causes, a phenomenon that sustains and expands the HIV pandemic year by year. Many of us have devoted our professional lives to combating these numbers. And those efforts have led to great progress in HIV prevention in recent years, as you've heard earlier in this meeting. Yet, considerable risks, including demographic changes and other competing global priorities, threaten this progress. Recognizing these threats, UNAIDS observed just last year that, as much as ever, the world needs an efficacious HIV vaccine. UNAIDS' observation was based, among other things, on data and models like this. Here, the steeper decline in the dotted lines in this graph reflects the addition of an HIV vaccine to supplement other HIV prevention tools and approaches. In these models, it is only with the addition of an HIV vaccine to the prevention package that we see a reduction in new HIV infections below 500,000 per year. We even approach zero. Now, this model is in a low and middle income country context. But a similar phenomenon of substantially improved impact on HIV incidence and prevalence in the presence of an HIV preventative vaccine is observed in locations like Seattle, Washington, where I am based, where this model incorporates high access, uptake, and adherence to other prevention tools like PrEP. So that begs the question of what is stopping us from adding an HIV vaccine to this prevention package. This virus makes it difficult. As a side note, and by comparison, most of the challenges listed here contrast starkly with the lesser yet still considerable challenges posed by SARS-CoV-2. And if that's of interest, in the panel discussion, we can explore this contrast and consider the myriad ways in which HIV and COVID-19 prevention efforts have informed each other in the panel discussion. So HIV is a rapidly integrating retrovirus. It leaves only a very brief window, on the order of hours to days, for the recruitment of vaccine-mediated responses. It is also a hypervariable and evasive virus, and it infects humans. There's no ideal non-human model from which we can learn. It can be acquired across different routes and throughout the life cycle. Furthermore, for most of the last 4 decades since HIV was identified, it did not appear that HIV could induce protective immunity. At least any protective immunity that it could induce was outpaced by viral evolution and escape. But with respect to this challenge, we've made significant progress in the last few years, particularly with respect to identifying a correlate of protection, which acts as a goalpost, if you will, for HIV vaccine development, and a validated marker that will help us understand how near or how far we may be from that goalpost with a candidate HIV vaccine or other biomedical prevention strategy. That goalpost or marker of HIV protection potential is related to neutralizing antibodies, antibodies that bind to the HIV GP120 envelope protein that is used by HIV for viral entry into host cells. And when these antibodies bind to GP120, they neutralize HIV, draining it of all color, so to speak, and preventing it from establishing infection. That process is reflected in this video, in which at the bottom of your screen, you see the host cell, like a CD4 positive T cell, with its receptors displayed on its membrane. And you see antibodies in circulation. These could be antibodies passively infused or elicited by a vaccine. When HIV comes into the picture, those antibodies bind to that spike protein on the virion, again, draining it of all color to represent neutralization, and HIV exits stage right. The use of neutralizing antibodies in this way is not new. Since the late 1800s, neutralizing antibodies, initially as part of serum therapy, and then as gamma and immune globulin, convalescent plasma, and now monoclonal antibodies, have been used to prevent infection or to prevent disease caused by many different pathogens. And most effective vaccines induce neutralizing antibodies to the pathogen. For years, scientists have been asking whether and or how HIV could be added to this table, to this list. And in 2016, the NIAID HIV Vaccine and Prevention Trials Networks, in collaboration with the Vaccine Research Center at the NIH, set out to explore that question of whether HIV could be neutralized by these neutralizing antibodies in that process. And they asked this question through two parallel global HIV antibody efficacy trials that were conducted at nearly 50 clinical research sites worldwide. The trials evaluated a monoclonal antibody called VRC01, which binds to the HIV GP120 spike protein at the locations depicted here in red, the CD4 binding site on the HIV envelope trimer. Over 4,600 participants, including men who have sex with men and trans individuals in the Americas and Europe, and women in sub-Saharan Africa, enrolled and received either lower or higher dose VRC01 or placebo, and they received this in an IV infusion that was given at enrollment at week 0, as depicted here, and then every other month thereafter for a total of 10 infusions administered over about 2 years. HIV diagnostic testing was conducted at least monthly, and a full HIV prevention package was available to all participants. The AMP trials proved the concept that yes, HIV can be effectively neutralized. That is, it may yet be able to join that list of pathogens for which infection or disease can be prevented by neutralizing antibodies. But the degree of neutralization necessary to prevent HIV acquisition was only observed in individuals who were exposed to viruses sensitive to the AMP VRC01 mAb. So we learned about the breadth and neutralization potency necessary to prevent HIV infection. And importantly, this neutralization was detected with an assay that can now serve as a validated tool to predict efficacy of other preventative HIV broadly neutralizing antibodies and of the vaccines designed to elicit them. Finally, the neutralization titer readout from this assay appears to have provided us with that elusive correlative protection. And thanks to the AMP trials, we now better understand how to correlate the neutralization that we observe in non-human primate models to humans. Altogether, these advances are really quite transformational for the global effort of efficient HIV vaccine and monoclonal antibody development. However, overall efficacy in the AMP studies was not observed, with positive efficacy point estimates, but confidence intervals clearly crossing 0, as depicted here. That leads us to ask, what is available for the majority, depicted in the lower 2 lines of this graph, who were not sufficiently protected from infection by VRC01? Well, VRC01 is now one of many available mAbs against HIV. Several other mAbs binding to a similar area of the GP120 at the CD4 binding site are in clinical trials, and several mAbs that bind to other neutralization-sensitive epitopes, like the V2 or V3 regions of the envelope protein, are also in clinical trials. To illustrate the implications of these numerous options for HIV neutralizing antibodies, let's look at a familiar analogy. On the left of the screen is a depiction of the HIV replication cycle in a target cell, with various stages in that cycle labeled in black. On the right is a depiction of the HIV GP120 with various neutralization-sensitive epitope regions, like the CD4 binding site that we were looking at earlier, and others labeled here in different colors. Different antiretrovirals target different points in the HIV replication cycle, just as different monoclonal antibodies depicted here target different neutralization-sensitive epitopes or locations on the HIV envelope protein. We know the impact that combination ARVs can have in support of optimal HIV treatment, and we hypothesize that combination mAbs can have a similar impact in support of optimal HIV prevention. mAbs, like antiretrovirals, target the structure of HIV, the HIV envelope protein, in order to impact the function of HIV, i.e. to neutralize the virus, preventing GP120-mediated viral entry and the consequent establishment of HIV infection. As with antiretrovirals, utilizing mAb combinations may limit the acquisition of mAb-resistant viruses and may limit in vivo viral escape if HIV is transmitted in the presence of mAbs. These trials here listed from the HVTN, CAPRISA, and the Rockefeller Group are all evaluating combination monoclonal antibodies for HIV prevention. In addition to continued evaluation of mAb combinations, there are several other ways in which the HIV monoclonal antibody field is advancing, such as through identification and development of additional antibodies, including those with greater neutralization potency and breadth. Engineering of the FC or foot portion of mAbs to extend half-life and to enhance additional antibody functions like antibody-dependent cellular cytotoxicity or phagocytosis, ADCC or ADCP, to supplement the neutralization function. We're also working on improving mAb delivery. For example, appreciating that while IV infusion was highly successful in the AMP studies, subcutaneous delivery can be much less challenging in many settings. And last but by no means least, working with community, industry, and other stakeholders to prepare for global implementation of this HIV prevention approach. Even with much more ground to cover with mAbs for HIV prevention, they've already contributed greatly to our efforts to address many of the challenges of HIV vaccine development, as listed here. And if you'd like, we can explore these further in the panel discussion. The HIV vaccine effort was clearly ready for this kind of help. While no HIV vaccine approaches entered efficacy assessment for the first 2 decades of the HIV pandemic, in the last 2 decades, over half a dozen HIV vaccine efficacy trials have launched. In each trial, various viral components have been included in vaccine inserts that were formulated into DNA plasmids, viral vectors, or as protein administered with various adjuvants, and each regimen generally intended to stimulate one or both adaptive immune arms, eliciting B and or T cell responses. Only one of these trials has yet demonstrated efficacy. In the RV144 Thai trial, 60% vaccine efficacy was observed at 1 year, declining, however, to about 30% at the pre-specified primary efficacy time point a couple of years later. For today's update, I'll speak a bit more about the 2 most recent HIV vaccine efficacy trials listed here, collaborations between the HVTN and Janssen. The MBOKODO trial, as well as the MOSAICO trial that I'll mention in just a moment, were informed by 2 non-human primate experiments in which a viral vector and protein vaccine regimen provided a high level of protection, over 90% protection from infection per exposure in monkeys, and a phase 1 trial demonstrated evidence of encouraging broad immune responses elicited by the vaccine regimen in humans. The MBOKODO efficacy trial sought to extend these findings to demonstrate HIV vaccine efficacy, prevention of HIV infection, in women in sub-Saharan Africa. The vaccine regimen consisted of an adenovirus shell that contained a mosaic vaccine insert, a blend of GAG, POL, and OMB amino acid sequences from multiple clade B and clade C viruses. And that was designed to elicit the broad cross-clade immune responses that were observed in the phase 1 trials. That mosaic viral vectored vaccine was given at 4 time points over the course of a year. And then at the last 2 time points, a clade C protein vaccine was co-administered with the mosaic viral vectored vaccine. The vaccine regimen in MBOKODO was safe, with no differences in moderate or severe adverse events across treatment arms, vaccine or placebo. However, the regimen did not prevent HIV infection, with a point estimate of 25% vaccine efficacy, and again, a confidence interval that crossed 0 at the pre-specified primary analysis time point 1 year after participants completed vaccination. The ongoing MOSAICO trial, which is being conducted among MSM and trans individuals, utilizes a very similar regimen to that evaluated in MBOKODO. The primary difference is that in MBOKODO, at the last 2 boost vaccinations, an additional mosaic protein-based vaccine was co-administered with the last viral vectored mosaic and clade C protein vaccine. And that, again, is intended to add further breadth for a truly global vaccine. This study is ongoing in 8 countries throughout the Americas and Europe. So where are HIV vaccine efficacy trials going from here? To come somewhat full circle to start, the neutralization-based biomarker that was validated in the AMP studies can help us down-select more efficiently from early to later phase trials, and can help us conduct later phase trials more efficiently through interim analyses that may reveal neutralization dynamics and thus prevention potential before an efficacy or non-efficacy signal may be available. The role of other HIV prevention modalities that you've heard of in this meeting must also be considered in the design of HIV vaccine efficacy trials. This is a topic to which whole meetings are devoted, and I won't explore it in greater detail here, but we can do so in the panel discussion if you'd like. The HIV vaccine field, with pioneers like Caroline Carico and Drew Weissman, greatly aided the COVID-19 vaccine effort by sharing over a decade of expertise that they had built in mRNA technology in the HIV vaccine field largely. The resultant success with COVID-19 was inspiring and feeds back into the HIV vaccine field, as I'll outline a little bit more in a moment. The HIV vaccine efficacy trials that I showed just a few moments ago from the last 2 decades have relied primarily on an empirical approach to HIV vaccine design. But rational design of HIV vaccine immunogens is leading the way in the next generation of HIV vaccine iteration, as I'll touch on also just briefly in a moment. And last but by no means least, authentic and early engagement with communities to lay the foundation for successful HIV vaccine implementation is critical. This is another area in which the HIV vaccine field's experience informed COVID-19 vaccine development and implementation and rollout. It is also an area from which the HIV vaccine field can, in turn, learn from the successes and challenges with COVID-19 vaccines. One thing that is not entirely clear is which approach to an HIV vaccine may be the best to advance in the next efficacy trials. DNA, viral vector, protein, mRNA, and various combinations of all of these have been tried, and they have different advantages and disadvantages. We see this in the COVID-19 vaccine field as well, where mRNA vaccines were among the first to launch and have truly been a shining star. But viral vector, protein, and combination vaccine regimens have also clearly had a place in the global armamentarium. There are advantages, we think, to continued diversification in this regard. I do want to spend a moment in this overview of HIV vaccines to highlight a few approaches that are up and coming. The first is a reverse engineering approach, the idea of taking broadly neutralizing antibodies identified in individuals who've developed them over years of living with HIV and mapping where on the HIV envelope protein those antibodies bind. Then a copy of that bNAb target, that neutralization-sensitive epitope on the GP120, can be made in the lab and then given in a vaccine in order to stimulate the immune system to make those bNAbs even before the immune system has ever been exposed to HIV. There are several early phase HIV vaccine trials, including those from the HVTN and IAVI that I've listed here, evaluating this concept. 2 other rational vaccine design approaches include germline targeting and lineage-based approaches. The idea for each of these approaches is to essentially recapitulate through sequential immunization the process that occurs in perhaps 10 to 20% of individuals chronically living with HIV. In this process, these cells evolve from germline through affinity maturation and somatic hypermutation to plasma cells that secrete bNAbs. Several early phase HIV vaccine trials are evaluating these approaches as well. Finally, a major rate-limiting step to advancing promising HIV vaccine candidates is the reliance on recombinant protein technologies for which years may be required to move from immunogen to clinic. mRNA technologies can, however, facilitate rapid iteration. We are getting a glimpse of the potential of mRNA technology in COVID-19 vaccine development, and several early phase trials are launching now to evaluate this in the HIV vaccine field. All of these approaches to HIV vaccine development rely heavily on collaboration. That includes, as depicted here, collaboration across multiple fields of scientific expertise and multiple immunologic approaches. It also includes collaboration across many stakeholders, including, importantly, community members and trial participants, public and private partners, industry, and academia. This also serves as my acknowledgement slide, as I am indebted to representatives from each of these bodies and many others for this overview of HIV vaccines and mAbs. As we proceed to the panel to explore the question of whether we can ever control HIV without a vaccine, I'll add thanks for one individual in particular and highlights of some wisdom he has shared over the years. I think we now know that no single tool is likely sufficient to prevent HIV and AIDS globally. That's been our experience with SARS-CoV-2 and COVID-19 as well. However, there is a single tool, an HIV vaccine, that while perhaps not sufficient, may well be a necessary component of any HIV prevention strategy that hopes to truly end HIV and AIDS once and for all. I look forward to talking with you further in the panel. Thank you.